Okay, so moving on. Uh, let's talk about the hard part. Let's talk about what's actually happening technically, what's physically happening here. So, this is probably the biggest word in honors biology. It's called photophosphorylation, but don't let it be scary because you already know what phosphorylation is. Phosphorylation is just adding a phosphate group to something. What we're trying to phosphorylate is ADP. We want to phosphorylate ADP, which means adding a phosphate group to it so that it becomes ATP. Because if you remember the ATP cycle, ATP is the high energy version of ADP. So the goal is to phosphorylate ADP. At least that's one of the goals of the light-dependent reaction. The other goal is to make NADPH. So in photophosphorylation, the ETC pumps hydrogen ions into the thylakoid space. When we say that we're exciting the electrons in PS2 so that we can use their energy to do something, technically what we're using that energy for is this transmembrane protein. It's called the cytochrome complex, actually a big giant protein with a bunch of stuff in it. But basically what it's doing is it's active transport. It's pumping hydrogen ions into the lumen. And they don't want to go into the lumen because the concentration in the lumen is already high. So we have to use some energy to push these hydrogen ions into the lumen to build that concentration gradient. Now, I haven't actually told you how this makes ATP yet, but you might have already guessed it. As we build up this concentration gradient of hydrogen ions, well, what do particles in the universe want to do? They want to go down their concentration gradient. So the hydrogen ions, they actually want to leave the lumen. The problem is they can't leave through the ETC, and they can't leave through the photosystems because they are polar, okay? They have a charge. So they can't just easily go through the phospholipid membrane. They have to go through some type of channel. Well, that's where ATP synthase comes in. ATP synthase is a super cool membrane protein enzyme that helps to make ATP. And how it does this is actually when the hydrogen ions go down their concentration gradient, they actually go through this protein here, ATP synthase. It's an enzyme. And as they go down their concentration gradient and out of the membrane, it actually causes ATP synthase to spin. And as it spins, it phosphorylates ADP into ATP. So we build up the concentration gradient of hydrogen ions so that as they pass down their gradient, they spin the ATP synthase and they make the ATP. This is why we pump them into the lumen. You pump them into the lumen to build the gradient. Okay? If we build that gradient, this is going to power the ATP synthase. So we actively transport hydrogen ions into the lumen, and then we harvest that concentration gradient energy to spin ATP synthase and phosphorylate ADP. Now, there's actually another source of hydrogen ions that's contributing to the concentration gradient, and that's the splitting of water. Because remember, as the electrons leave PS2, we can say that PS2 is oxidized because PS2 loses its electrons. So to replace those electrons, we split the water in the lumen so that the electrons can replace the ones that were lost. We reduce PS2 by splitting water. But when you split water, you have an interesting result. You have oxygen gas, which is small and nonpolar, so the oxygen gas just diffuses out, and that's why plants release oxygen gas, okay? And that's good for us because we can breathe it. But the hydrogen ions, they can't leave so easily because they're charged. So the hydrogen ions instead are just going to float in the lumen and build up with this concentration gradient until they can flow through the ATP synthase.